Hello, you are most welcome, ladies and gentlemen, to Real Impartation Moment on Tuesday Night Anatomy with Daniel Okwan. On today's section, we want to look at the histological organization of the tubular organs of the post-pharyngeal gut. Yes, for tubular organs, generally they will have three coats or three tunics, namely from the lumen to the external. Yes, we are going to have what we call mucosa, muscularis, and of course adventitia or serosa. But for tubular organs of the post-pharyngeal gut, yes, the alimentary canal, we are going to have four tunics or four coats. And it's because, yes, there will be an additional coat, which we call it the submucosa. So instead of having three coats, it's going to have four, namely mucosa, submucosa, muscularis externa or muscularis propria, and of course adventitia or serosa. And of course, we're able to distinguish, between, the submucosa from the mucosa with the, presence of what we call the muscularis mucosae. So muscularis mucosae, which is actually smooth muscle, will be an important identifying feature for the organs of the post-pharyngeal gut, apart from having this kind of four-core system I'm talking about. So without much ado, yes, let's quickly run through this system that we're talking about. So generally, yes, we know that for the organs of the gastrointestinal tract, yes, we have what we call the main organs. Okay, so for instance, we have the oral cavity, structures in the oral cavity, the mouth. Yes, we go through, of course, the pharynx, the laryngopharynx, oropharynx, eventually into what we call the esophagus. Esophagus into the stomach, into the small intestine, eventually into the large intestine. And of course, those ones which have to be absorbed, will be absorbed into the bloodstream. And then the undigested food material, yes, will have to be ingested through, of course, the anal canal. So essentially, yes, we know that the mouth, yes, will contain teeth, will contain salivary glands and all that. Yes, will contain a tongue to roll the food into a bolus, okay? In fact, the saliva will initiate a chemical digestion, okay, in, of course, the food. Of course, yes, through swallowing in the pharynx, yes, gets into what we call the esophagus. Esophagus will propel the food from the laryngopharynx into, of course, the stomach. The stomach will be a temporary reservoir for this kind of bolus of food, okay, to also, churn this food mechanically into chyme, okay, and then with the help of several cells, okay, which it has, for example, the parietal cell secreting what we call hydrochloric acid. Yes, this kind of secretion which will be coming from the the stomach, yes, mixes with the food, form what we call chyme. Okay, yes, continue with the chemical digestion of food, yes, with the help of course, pepsinogen produced from what we call chief cells. Yes, we see all these ones will be by some of the functions that the stomach will perform. Yes, of course, mainly the chemical digestion, yes, will continue in the duodenum. Yes, most of the absorption will take place in the jejunum and ileum. Okay, yes, in there, everything will happen over there. Yes, the large intestine reabsorbing a lot of water from the undigested food materials, as well as reabsorbing electrolytes, including vitamins from the undigested food material. Yes, of course, rectum will compact the fecal matter. Apart from that, yes, it will also help store, okay, this fecal matter temporarily before it is, ingested through the anus. So these are some, things that we have to know. Then you know that we also have some accessory organs of the GIT. Accessory organs talk about, the teeth, talk about the salivary glands, talk about, the liver, talk about the gallbladder, okay, talk about the pancreas. These are all accessory organs which are going to play important role by way of some secretions, okay, which is going to help in ensuring that digestion, is efficient. Yes, roughly, if we take from the oral cavity all the way to the anal canal, the digestive system will roughly, measure, 9 to about 10 meters. Okay, so let's move on and see. So by way of function that this digestive system is going to play, yes, it will include ingestion, and that's taking food into the body through the mouth. Okay, yes, will be by way of secretion. Yes, several secretions rich in enzymes, rich in other, either being alkaline or acidic, okay, to help in actually ensuring that digestion is efficient. Yes, there will also be what we call propulsion. Okay, propulsion will be very important by way of transporting the food product from one part of the gut to another. Importantly, digestion, which will include mechanical digestion, okay, without the action of enzymes. For instance, chewing, talk about mastication, talk about churning of food in the stomach. Yes, by way of chemical digestion, then we want to convert that food product into another, with the help of, digestive enzymes. Then after all, yes, after the end product of digestion, for instance, if it's carbohydrate food, mainly, yes, glucose, talk about, protein food, mainly, amino acids, talk about, these lipids, fatty acids and glycerol, yes, they will be absorbed into what we call the bloodstream, or they will be absorbed into what we call the lacteals, okay? So we see that. Then of course, defecation by way of, the undigested food material getting eliminated out of the gastrointestinal tract through the anus. And these are what we call the main functions that the gastrointestinal tract, the GIT, is going to perform. But this time around, we are interested in the part of the GIT which is below the pharynx, below the laryngopharynx, and that's the post-pharyngeal gut. So let's see what we have. So we have a general histological organization or plan for the organs of the post-pharyngeal gut. Now, once we are having a tubular organ, every tubular organ will have a lumen, a central compartment, where the substances will have to go through. And in this case, here is the lumen, the opening in there, the central cavity over there, that's the lumen. Plural is lumina. And then from the lumen to the external environment, external aspect, we are saying that it's going to have four tunics or four coats. And these tunics are, yes, what we call mucosa. Then beyond the mucosa, we have submucosa. Then we have what we call muscularis externa. Then we have what we call adventitia or serosa. Now, if we talk about the mucosa will be made up of two main things generally. But for the post-pharyngeal gut, it will be made up of, four things. Now, if it is two, then it will be made up of a surface epithelial lining, okay, surface epithelium, and then an underlying lamina propria of loose connective tissue. But for the post-pharyngeal gut, there will be a third layer which will contribute to this, and that is what we call the internal muscle layer. And this internal muscle layer is what we call muscularis mucosae. It's the muscle of the mucosa, exclusively smooth muscle. Then we have what we call the submucosa. Now, the lamina propria is made of loose connective tissue. The submucosa is also connective tissue, but this time around it is dense, irregular connective tissue. Submucosa here is dense, irregular connective tissue. We will see other things that I'll be making mention of, okay, in these areas. Then we also have what we call muscularis externa. Now, generally, for other, tubular organs, we will have what we muscularis. We don't qualify to the externa or propria. But for the organs of post-pharyngeal gut, we will qualify them with the word externa or propria. The reason is that, yes, because we already have a muscularis interna or muscularis mucosae as part of its mucosa, and therefore we have to qualify, the proper muscle or the external muscle. Now, this external muscle will be made up of what we call inner circular layer as well as, outer longitudinal muscle layer generally. But we get to know that for the stomach, it will, because of its churning ability, its churning function, then what will happen is that it will be made up of three layers: inner oblique, middle circular, outer longitudinal. We will see those ones. Then we have the outermost tunic. Now, for the outermost tunic, yes, it is either serosa or adventitia. Okay, it's either serosa or adventitia. Now, if it is serosa, then we have what we call connective tissue, actually loose connective tissue, surrounded externally by a single layer of mesothelium. That's simple squamous epithelium, and that makes it, what we call serosa. But if there is no simple squamous epithelium lining the loose connective tissue, but rather the loose connective tissue adheres to surrounding structures, then we call it adventitia. So we see all these ones. So that is what we are going to see. Now remember that because the food is coming from the external environment into the body, then what will happen is that, in the mucosa, we will have what we call, some kind of lymphatic tissue or lymphoid tissues will be endowed in there in the mucosa, even as well as in the submucosa, and that is what we call lymphoid organs. And we see malt and gall, we see all these ones. So yes, it's not surprising that if it is serosa, yes, by way of having a double fold of peritoneum connecting to the posterior abdominal wall, if it's coming from the GIT, yes, for instance, in the organs of the abdomen, intraperitoneal organs will have serosa, and therefore those serosa will be modified into what we call mesentery proper. For instance, if you have this coming from the small intestine, yes, connecting to the posterior abdominal wall, then these blood neurovascular structures can enter, of course, to supply, you know, the various tunics of what we call the post-pharyngeal gut. So yes, I told you that we are going to have 4 tunics. Yes, from the lumen, the first tunic is the mucosa. Now mucosa is having 3 components. The first component is the epithelial lining. Now the epithelial lining, yes, is going to be, I mean, look at it. You see that I've included even the mouth, that of the pharynx, actually that of the laryngopharynx or oropharynx. Remember that nasopharynx will be actually pseudostratified columnar epithelium with goblet cells and cilia. Okay, will be ciliated, having goblet cells, nasopharynx. But for oropharynx and as well as, you know, laryngopharynx, yes, they will have what we call stratified squamous non-keratinized epithelial tissue. Now for these wet surfaces which are getting to the external environment, they will have stratified squamous, you know, non-keratinized epithelium. Then the remaining components, yes, the stomach will have simple columnar epithelium without goblet cells. Very important. Please note that it is without goblet cells. For small and large intestine, they will have simple columnar epithelium with goblet cells. Anal canal, because it's getting to the external environment, will be stratified squamous non-keratinized epithelium. So in a simple sentence, what we can say is that the organs of the post-pharyngeal gut, yes, the epithelial lining, will be, with the exception of the esophagus and the anal canal, having what we call stratified squamous non-keratinized epithelium. The remaining component of the organs of the post-pharyngeal gut will have what we call simple columnar epithelium with or without goblet cells. The only one without goblet cells is the stomach. So there we are. This is the epithelial lining of what we call the esophagus as well as the anal canal, because both of them will have stratified squamous non-keratinized epithelium. Now it is stratified because you can see several layers of cells. Epithelial. Now for you to know that this is the epithelium, you can see that the epithelium stains quite deeply stained, stains intensely compared to its underlying lamina propria of loose connective tissue. Lamina propria is lightly stained. So this stratified squamous, okay, so it is actually stratified. And then you look at the most apical cell shape. You can see that the most apical cell shape is actually squamous, okay. Now another thing too that you can help you under clue for you to know that we are looking at stratified squamous epithelium is that whether it's keratinized or non-keratinized, is that you see that the epithelial lining, the epithelium dips into the connective tissue area. Okay, so the epithelium dips downwards into the connective tissue area, and this is what we call epithelial pecs or epithelial ridges. Now these epithelial pecs, yes, in turn, yes, the underlying connective tissue also, you know, invade the area of the epithelium. Okay, and that is what we call connective tissue papillae. Okay, plural is connective tissue papillae. Okay, so by virtue of seeing epithelial pecs and connective tissue papillae, then we know that we are looking at stratified squamous, you know, epithelium. In this case, because there's no non-cellular layer of keratin here, then we say it is non-keratinized. Now the only area that we tend not to see these epithelial pecs and connective tissue papillae, you know, is in the cornea. Okay, so that's what we see. Now one thing is that yes, for the epithelium of the esophagus as well as the anal canal, you see similar architecture. But one thing that we will find is that, yes, very soon we get to see is that for the esophagus, you're going to have a prominent muscularis mucosae. But in the anal canal, yes, we don't see muscularis mucosae because beyond the anorectal junction, we don't see muscularis mucosae. So if we are having 2 histological sections, both showing stratified squamous non-keratinized epithelium, actually from the post-pharyngeal gut, and we are asked to distinguish, yes, for you to know that this is the esophagus or this is the anal canal, yes, look out for the presence or absence of muscularis mucosae. You don't find muscularis mucosae in the case of the anal canal. Yes, we get to the stomach. I told you that the epithelial lining of the stomach is simple columnar epithelium. Okay, so you can see that the cells over here are columnar shaped. This is the epithelial lining. Okay, they are columnar shaped. Okay, you can see they are elongated. The nuclei are elongated this way. okay, without goblet cells. Now the key word here is that it's without goblet cell. But the point is that we know that the stomach, secretes, this kind of mucus, okay, to line or to cover its, wall, so that it doesn't, suffer from auto digestion from its own secreted acid. So what happens is that yes, the stomach indeed secretes mucus, but this mucus is not coming from goblet cells because it doesn't have goblet cells. Therefore, what happens is that the surface epithelial lining of the stomach, okay, actually secretes what we call mucus. Remember that mucus is a mixture of water and mucin. So this area, okay, the columnar cells of what we call the stomach will secrete what we call mucin, and then it combines with water to form mucus. So you can see that apical. So this is the nucleus. These are the nuclei. Apical to the nuclei, this is the cytoplasm. You can see that the cytoplasm appears, mucinous, okay, appears somewhat pale. This area is filled with mucinous, substance, okay, it's filled with mucinous substance, and therefore, it will be PAS, positive, okay, periodic artery shift, positive, okay, when it's stained with it, because over here you have a lot of mucinous substances, okay, so bear in mind. Now, for both small and large intestine, yes, we are going to have, you can see these cells. Now, this cell is a goblet cell. You can see this area is pale. This is the nucleus, as if there's a wine glass. Okay, these are goblet cells interspersed within the columnar cells, okay, of its epithelium. So you can see that we can have numerous. You can see these pale staining areas. Okay, these are all goblet cells. Okay, yes, both small intestine and large intestine all have goblet cells. But what happens is that as you move distally, okay, from the small intestine down, okay, from the duodenum, jejunum, ileum down to what we call the large intestine, then what happens is that the number of goblet cells, okay, population begins to increase, okay, increases along the length of the intestine. So that's what you have to know. Now, another thing too is that yes, for both small and large intestine, in its epithelium, you have what you call a brush border. I'll show you the brush border. So now, one other thing too that you have to know regarding yes the mucosa is that there are some special features, okay, that we have to know. Now, I told you that the epithelial lining for the mucosa, lamina propria is the same. Lamina propria will be loose connective tissue, areola. Okay, now in the lamina propria you may or you may not find glands, so or you may find certain peculiarities. I told you that you may see certain lymphatic nodules or you may see diffuse lymphatic nodules, okay, which is a common thing, but there are some areas of special interest. So for instance, if you look at the esophagus, generally doesn't have what you call mucosal glands, okay, in the lamina propria, except at its distal end. So esophagus is devoid of mucosal glands except at the distal end, and that's almost the area which is related to the abdominal area, because that area is related to, of course, the stomach. Okay, it's closer to the cardia of the stomach, and therefore that area, is endowed with what we call esophageal cardiac glands. I'll show you. Now the stomach, yes. Now one other thing too that I have to tell you is that yes, another area which is devoid of mucosal glands will be the villi. I'll show you the villi. Okay, so the villi, as well as the proximal part of the esophagus, will be devoid of mucosal glands. Now the stomach, yes, will have what you call gastric glands. Okay, I'll show you with its ducts known as gastric pits. Okay, then small intestine, yes, will have what you call mucosal glands, okay, in the lamina propria known as crypts of Lieberkuhn. The same applies to the large intestine. So if I talk about large intestine, I'm talking about the cecum, ascending transverse, descending sigmoidal colon. Even the rectum, okay, will be part of it, as well as even the appendix, okay, will all have what we call crypts of Lieberkuhn. Then we also have what we call the ileum. Ileum will have what we call Peyer's patches. I'll show you the Peyer's patches. Peyer's patches will be exclusive to the ileum, located actually at the antemesenteric border of the ileum. Then, of course, for appendix will have a peculiar architecture, which we describe as having circumferentially, arranged, lymphatic nodules. So there we are. Now I want to show you the cardiac glands. Now look at this. Now here we are having what we call the esophagus. Okay, so at the point where the esophagus is about meeting what we call the stomach, we have what we call cardio-esophageal junction. Cardio or gastro-esophageal junction, because it's going to meet the cardia, we say cardio-esophageal junction. So just around that area, okay, we have what we call mucosal glands for what we call for the esophagus. Okay, and that one is in the lamina propria because it's mucosal. Okay, and it will secrete what we call neutral mucus. Okay, so that it protects the lower portion of the esophagus from the acidic content, that chyme that may reflux in there. Okay, so that's what we are going to see. Now again, for the mucosal glands for the stomach, I told you that the stomach is going to have this gastric gland. Okay, and this gastric gland will have a duct and it's known as the gastric pit. Okay, and this is the neck of the gastric gland. Okay, now over here we are going to have the surface mucous cells. So the stomach is going to have some cells lining the surface, and that one will secrete mucus, and that's those ones that we've seen already, and that is what we call the surface mucous cells. Okay, or mucous surface cells. Then we also have what we call mucous neck cell. Around the neck of the gastric gland, we have mucous neck cell. So surface mucous cells and mucous neck cells, yes, will actually be secreting what we call, mucus. But what happens is that the more you are getting close, now these ones will secrete, it's going to be rich in what we call bicarbonate. Okay, the mucus that they will secrete will be rich in bicarbonate, so that by way it will tend to neutralize the acidic content of the chyme actually. Then at the upper half of what we call the gastric gland, yes, we have these eosinophilic cells which appear like fried eggs. Yes, usually that's how we describe it. Okay, having fried egg appearance, we call them parietal cells. So they are acidophilic or eosinophilic. And then these parietal cells, yes, mainly what they are going to do is that they are going to pump, or they are going to produce what you call hydrochloric acid. Yes, by actually pumping hydrogen ions and combine it with chloride ions to form what you call hydrochloric acid. Remember that hydrochloric acid will be very important as far as, of course, activating pepsinogen, yes, to pepsin. Yes, very important in actually, yes, stopping the activity of salivary amylase in the stomach. Yes, it will be very important in actually, yes, killing most bacteria or pathogens in the food that you've taken in. Yes, it will also be relevant in actually, yes, breaking the connective tissues in actually meat. So these ones will be some of the functions that the hydrochloric acid, yes, will perform. But in addition, these parietal cells also secrete some kind of factor we call intrinsic factor. Now, intrinsic factor is very important because it is needed in the terminal part of the ileum for absorption of, vitamin B12. It's not calcium absorption, it's actually vitamin B12. And vitamin B12 is what you call cobalamin. So cobalamin absorption is very important over there. And that is why, because vitamin B12 is needed, okay, in the bone marrow for erythropoiesis, production of, red blood cells. Otherwise, it will result in what you call form of macrocytic anemia we call pernicious anemia. So if someone goes through partial or total gastrectomy, then the reason why the person may suffer from some form of anemia. Now another cell which is worth mentioning, which is found in the lower aspect of the gastric glands, okay, usually found in the lower part of the gastric glands, they are actually basophilic, and those ones we call them chief cells. Now remember that parietal cells, we also call them auxintic cells. Now, chief cells, also known as peptic cells, yes, will produce what we call pepsinogen, which will be activated to pepsin to help in the chemical digestion of protein food. Yes, also gastric lipase, yes, will also be produced there, yes, to help in lipid digestion, chemical digestion. In addition, yes, we also have some kind of cells, we call them enteroendocrine cells, which are going to make some kind of hormone-like substances to control the activity of the gastrointestinal tract. And these hormone-like cells, yes, we call them enteroendocrine cells. Examples include the D cells. Okay, the D cells will produce what we call somatostatin, okay, which will inhibit the activity of parietal cells in secreting hydrochloric acid. Okay, Then of course we also have what you call G cells. Okay, so G for gastrin. So G cells will produce this hormone-like substance known as gastrin, which will stimulate the parietal cells to make acid-rich products. Okay, gastric secretion. Okay, so that is what we have to know. Now, talking about the stomach, yes, I've told you that the stomach will have what we call simple columnar epithelium without goblet cells. Although yes, the columnar cells will be secreting mucus. Then what will happen is that there's downward growth, okay, into what we call the connective tissue area, forming glands, and the glands will have what we call gastric pits, and these pits will serve as the conduit for conveying the products onto the surface of the stomach, surface line of the stomach. Then the secretory units, which we are calling them glands, okay, are these ones. So one thing that you have to know is that with respect, okay, to the proportion of the pits, gastric pits to gastric glands ratio, the secretory units can help us to know, which area of the stomach we are talking about. Although grossly we know the stomach is going to have what you call the cardiac part, yes, the fundic part, the body or the corpus part, as well as the pyloric part. What we are saying is that for histologically we are able to distinguish between three main parts, and these three main parts are, yes, we are going to have what we call the cardiac portion. Yes, we are going to have, yes, body or fundus. So we are unable to distinguish between the body and fundus, okay, histologically, and then of course the pyloric part follows. Okay, so we can see that over here, you can see these over here, the upper portion. Okay, I told that the gastric glands mostly located in the upper portion will be the eosinophilic ones, known as oxyntic cells, or also known as the parietal cells. And then of course lower portion of the gastric glands, we are having the basophilic ones, which we call them the chief cells. So we see those ones. So there we are. Yes, we have actually the gland which is peculiar to the cardiac. We call the cardiac gland. The glands which is peculiar to, of course, the fundic or body part, which we call them, the fundic glands, or properly we call them gastric glands proper. Then we also have the pyloric glands, which is related to the pylorus of the stomach. Now remember that we are going to use the ratio. Now look at this, the pits, this is the ducts, this is the gastric pit, and this is the extent of the gland. So we're going to use the ratio of the pit to the gland ratio, pit to gland ratio, to tell the area that we are looking at regarding the stomach. But bear in mind that generally the gastric pit tends to increase, okay, along the length of the stomach, moving all the way from the cardia to fundus to body, yes, to pylorus. The pits, okay, in other words, the gastric ducts or the ducts of the gastric gland will be increases along the length. However, if we take a particular section of the stomach, for instance, if we take the cardia, what we say is that yes, the cardia is going to have almost the same length regarding the pits to, of course, the glands. Okay, so that's what we are going to see. And then usually, yes, the duct over here shows some kind of branching. Okay, so it may be simple, and then sometimes some will be straight. Okay, this one is straight without showing any branching, but this one is showing some kind of branching. Okay, in the case of the cardia. Okay, and then essentially, yes, to possess most of the cells over here, yes, essentially may have few parietal cells if they will be present. But chief cells are virtually absent over here, but you may see some parietal cells over here, a few of them. Then we go to the fundic part or the body part. What will happen is that yes, we are going to have the gastric pits will be just about a fourth to that of what we call the gastric glands. So we have less pits compared to the gland. Okay, so that yes, what we are saying is that for most of the cells that we are going to find in the case of the gland, okay, will be what we call, yes, we are going to have a lot of parietal cells. That's the key thing over here. We're going to have a lot of parietal cells. There will be chief cells over here. Now there was one cell that I failed to mention it to you. Now another cell which you find it in the case of the stomach will be what we call stem cells. Now get to know that stem cells are undifferentiated, okay, so they can give rise to committed adult cell types. So stem cells are located at the neck of the gastric gland, okay, the neck of the gastric gland. such that it is strategically positioned so that it can differentiate, okay, to give rise to the cells that are apical to it or give rise to cells that are basal to it. Okay, so another cell type which you're going to see in the case of, you know, the gastric gland, as part of the gastric gland content, yes, will be what we call the undifferentiated stem cells. Okay, so authentic cell, parietal cell, numerous of them will be found over here. Okay, you'll find all these ones over here. All right, now for the pylorus, for the pyloric part, what happens is that you have a very long duct. Okay, that's the pit. The pit is very long compared to that gland itself, the excretory unit. So we have long pit, short, you know, gland. Okay, for the pyloric part. So roughly we can say that yes, about 3 to 4 times the length of what we call the gland is the pit. And these ones, yes, for what we call the fundic part or the body, what we see is that yes, there will be simple, yes, they may show some kind of branching. Okay, simple and show some branching, simple branch, you know, actually tubular. Then for pyloric part, yes, they will show yes some kind of, you know, branching as well. Some may even show some kind of coiling, but rarely do we actually see these ones. So that is what we are going to see. But over here, you rarely may see what you call, parietal cells. You rarely may see, but you see a lot of, you know, chief cells around this area. Okay, so that's what we are going to see. So with these features, you should be able to distinguish between the various segments of the, the stomach. For instance, is cardiac portion, cardiac of the stomach. You know, almost the same ratio of pit to glands will be, yes, eventually the same. Then what we are saying is that essentially you may see some parietal cells, a few of them may be seen over here, but you won't see, you know, chief cells. Okay, with the glands, yes, showing some kind of branching, you may see some kind of branching, and then you may see some kind of coilings. Then or some may show just tubular without showing any branching. Then for the fundic part or the body, yes, we are going to see short ducts, compared to the gland. So that we have a lot of gland. Yes, the ratio is just about 1:4 between the pits and the gland. Okay, and then we are going to see a lot of what we call, parietal cells, authentic cells will be seen there. Yes, you may also see some, you know, chief cells. Chief cells will also be present. Okay, histological, mainly under light microscopy, these are the things we are going to use. Although at the electron microscope under special staining, which I will show you, yes, you may see all those ones. Then for pylorus, very long pits, short glands. Okay, then what happens is that you may see essentially chief cells with very few, if present, authentic cells or parietal cells. Now, we also have, as part of the mucosa, there will be some kind of glands which is made up of simple tubular, the actual simple tubular glands, and that is what you call crypts of Lieberkuhn. That's the mucosal glands, okay, which you find in the lamina propria of both the small and large intestines. Yes, that is what you call crypts of Lieberkuhn. Okay, so for crypts of Lieberkuhn, it's exclusive to what we call the small and large intestine, okay, in its lamina propria. But remember, although we know villi is related to what we call the small intestine, these crypts of Lieberkuhn are devoid or they are absent in the lamina propria of what we call the villi. Okay, please get to know that. So that is what we have. Yes, we know that the epithelial lining will be simple columnar. You can see these white areas. These represent what we call the goblet cells. Okay, and then we are going to see these cell types. Yes, these cell types are the absorptive cells or the enterocytes, which you call them the columnar cells. They are the simple columnar cells. In addition, you will have what you call these goblet shaped cells, wine glass shaped cells, which you call them, you know, goblet cells, will also be present, okay, in the surface epithelium. Then we also have what we call its stem cells. Now this time around, its stem cells is at the base of what we call its crypts. It's at the base of the crypts. Then just at the base too, you may see what we call Paneth cells. Okay, Paneth cells, okay, will secrete, will be rich in lysozyme. Okay, will secrete lysozyme. Okay, we've seen that it also has, you know, takes in a lot of zinc as well, and it has what we call antibacterial activity. Okay, fighting bacterial infections. Then we also have what we call the apude cells or the enteroendocrine cells, okay, which you call agantaphysin cells or agelophil cells. I'll show you because of this staining ability. okay, actually picking up the stain of what you call silver stains. Okay, so if you want to actually visualize what you call these enteroendocrine cells, then you have to use what you call silver impregnation method. Okay, so we see those ones. So these are the enterocytes. Yes, the enterocytes will bear what you call brush border, which I was showing you. Okay, brush border or striated border or microvilli. Now get to know that yes, there's a distinction I'll show you between brush border, striated border, and microvilli. If you are talking about microvilli, then it is electron microscopic structure we are dealing with. If it is brush border, usually, strictly speaking, it should be the proximal convoluted tubule of the kidney. But if you are talking about striated border, then under light microscopy, you are looking at it at the either the small intestine or the large intestine, and it's mainly for absorption, actually increasing absorptive surface area by a factor of 20. Then we also have what you call the goblet cell. Yes, goblet cell have a lot of mucinogen material which will secrete mucus. Okay, then we also have what you call the enteroendocrine cells. Okay, enteroendocrine cells, which will secrete, for instance, cholecystokinin, which will stimulate, of course, the gallbladder, okay, to secrete its, bile, or it will stimulate, the pancreas, to secrete its exocrine portion, and by way of secreting what we call, gastric secretion, and this gastric secretion, yes, will be less rich in what we call enzymes, okay, but secretin, okay, will ensure that it is actually rich in enzymes coming from the pancreas. Then the Paneth cells, we've talked about it. All right, so we are talking about the enteroendocrine cells. Now these enteroendocrine cells can also be called chromaffin cells or argentaffin cells. Yes, the reason is that yes, they are able to stain with what we call silver salts. Okay, so if you go through silver impregnation technique, then you're able to produce that. And then they produce what they call amines, including 5 serotonin, and then what will happen is that yes, we also call it, as a result, we also call it APUD, amine precursor uptake decarboxylase. Okay, 5-hydroxy tryptamine, 5-hydroxy tryptamine, so to produce these ones. And therefore we say that we also call them APUD cells, that's amine precursor uptake decarboxylase. Okay, and then these cells are positive to chromaffin reactions, and that is why we call them enterochromaffin or chromaffin like cells. Okay, so that's what we have to know. Now again, in the mucosa, another special feature that we find exclusive to the ileum is that, now look at this. Now this is the entire lamina propria or the mucosa of the ileum, but pertaining to just a side of it, of the mucosa, you can see these aggregated lymphatic nodules, okay, enlarged over here. Now you can see these are the finger-like projections. They are villi, which we see exclusive to, of course, the small intestine, including the ileum. So these ones are located at the antemesenteric border. So the ileum is actually intraperitoneal, connected to the posterior abdominal wall by way of mesentery proper. So that portion which is opposite, so if mesentery proper is attaching at this point, opposite to where the mesentery proper attaches is its antemesenteric border, and that is where you are going to see what we call Peyer's patches. Even Peyer's patches can be macroscopic as well. So under electron and light microscopy, that's how you see it. Peyer's patches, aggregated lymphatic nodules or cluster of lymphatic nodules located at the antemesenteric border of the ileum. Yes, also apart from that, we see in the case of the appendix, yes, some kind of lymphatic nodules, okay, which orient themselves or dispose themselves circumferentially in the mucosa of the appendix. Now, although yes, we are talking about yes, these things, restraining themselves in the mucosa, what happens is that as these lymphatic nodules grow, whether it's Peyer's patches, whatever, yes, or these circumferentially arranged lymphatic nodules, okay, what happens as they grow, they're able to break through the muscularis mucosae and enter the submucosa. So sometimes you may even see it in the submucosa. Now, talking about yes, Peyer's patches, talking about the circumferentially arranged lymphatic nodules, we also have the solitary ones, okay, because they are not really special. That's why I didn't make mention of. But what happens is that yes, we put all these ones together and we talk about what we call lymphatic tissue. We call them mucosal associated lymphatic tissue. Mucosal associated lymphatic tissue. But we don't have mucosa pertaining to only the post-pharyngeal gut or the GIT. Therefore, specifically for that of the GIT we are talking about, we should rather refer to it as gut-associated lymphatic tissue, which include the Peyer's patches we just seen, or the circumferential arranged lymphatic nodules we've seen, or the other solitary ones that we not made mention of. So you can find some in the bronchial tree or the pulmonary area, which we call a port or bolts. In the case of bronchial associated lymphatic nodule, yes, under the skin, you see what we call salts. Yes, you may see that in the nose, yes, not. You may see that in the larynx as LOLT, and then you may even see some in the conjunctiva as COT. So these are some areas that you may find, mucosal associated lymphatic tissue. It's not only exclusive to what we call the post-pharyngeal gut. That we find in the GIT is referred to as GOT, gut-associated lymphatic tissue. So I was talking about the three things. Yes, we've seen epithelial lining, we've seen the mucosa, the special things we can see in the mucosa. Yes, we also have what we call the third component of the mucosa. And the third component of the mucosa is known as muscularis mucosae or muscularis interna. And this muscularis interna is exclusively made of smooth muscle. It's exclusively made of smooth muscle. Generally, it is going to have two layers. And these two layers are what we call inner circular, outer longitudinal. Okay, but usually under light microscopy, the area that we tend to see these two layers a bit distinct will be the stomach. So usually under light microscopy, you don't see the two layers distinctly, except under electron microscopy. But for areas sometimes we see will be the stomach, where you can see the two inner circular, outer longitudinal. Again, as a result, we say that the muscular mucosa we find in the stomach is actually prominent histologically under light microscopy. Then also for the esophagus, it's also prominent, okay, to aid in that peristaltic movement, propelling the food, okay, to aid in actually what you call, bringing the luminal content closer, to the mucosa, okay. So that's what we are going to see. So what we are saying is that the muscular mucosa is going to also serve as the boundary between what we call the mucosa and what we call the submucosa, okay. That smooth muscle over there will serve as the boundary between the mucosa and the submucosa. But what happens is that this muscularis mucosa is very important in ensuring that, yes, the luminal content, in other words, the food products can be brought closer to the mucosa, so that of course the secretions and all that can actually act on the food products, okay. That's what we're going to see. Now, one thing that we see is that from the stomach, from the esophagus to the stomach, what happens is that then the muscularis mucosa begins to thin out. It becomes thin, okay, reduces in size until we get to what we call the anorectal junction. Now, beyond the anorectal junction, in the anal canal, muscularis mucosa is actually virtually absent. We don't find muscularis mucosa there. So that is what I want you to know. Now, we also have what we call the submucosa. Now, I told that the lamina propria of the mucosa is made of loose connective tissue. Now, for the submucosa, it's made of dense, irregular connective tissue. So you can see the collagen fibers actually oriented in different directions, okay, oriented in different directions, with actually, little ground substance, the white areas that you find over there, okay. Now, another thing that we find in the submucosa is that we are going to see what we call a nerve plexus. This nerve plexus actually, yes, gives some kind of innervation or supplies what we call the muscularis mucosae. So the muscularis mucosa is getting its nerve supply or its innervation from what we call this nerve plexus that we find in the submucosa, and that is what we call submucosal nerve plexus, and it's given the eponym Meissner's nerve plexus. Meissner's nerve plexus, and that is what I want you to know. Now, again, in the submucosa, you may see some special things over there. Now, the submucosa, generally, we don't tend to find glands. Now, we said that in the mucosa, in the lamina propria, we want to see glands except two areas. And those two areas, yes, we talked about, the upper portion of the esophagus, as well as what we call the villi. But for the submucosa, we don't tend to see glands. So anytime we see glands, then we qualify those glands with the word proper. So, for instance, we find glands. There are only two areas that we are going to see glands in the submucosa of the post-pharyngeal gut. and one is the esophagus. Now usually we talk about the upper half of the esophagus, but some books will say the entire esophagus. So the esophagus will have what you call submucosal glands, and these glands will be called esophageal glands proper. So if you hear of esophageal glands proper, then we are talking about the submucosal glands in the esophagus. Again, the duodenum will have what you call submucosal glands, and that one we call it submucosal glands proper. And it's also given the eponym Brunner's glands. Okay, Brunner's glands will also be seen there. But apart from these ones, yes, other than the Peyer's patches, I told you that in the ileum we are going to see Peyer's patches in the mucosa, although it's usually in the mucosa. But these Peyer's patches grow, then you're able to break through the muscularis mucosae to enter the submucosa. Now, so I want to show you the esophageal glands proper. Okay, the esophageal glands proper, yes, we are going to find it in the submucosa. So here is the epithelial lining. Okay, you can see these, epithelial pecs and connective tissue papillae, telling you that we are looking at stratified squamous, non-keratinized epithelium because there is no keratin over here. Then below it, there will be a layer of, lamina propria. Then there will be a smooth muscle layer, okay, separating them. Okay, separating them. Below that, we are going to have what we call submucosa. So within the submucosa, we are seeing glands, and these glands are actually compound tubuloalveolar glands, okay, or compound tubular glands actually, and these ones are what we call the esophageal glands proper. Now what are they going to do? Yes, they are going to actually secrete what we call mucus to actually neutralize the acidic chyme that may reflux from the stomach. For instance, if someone is suffering from GORD, gastroesophageal reflux disease, then that kind of neutralization can actually take place. So there we are. If you take the esophagus again, the epithelial lining, you can see the schematic diagram. You can see it's a stratified squamous. You can see the epithelial pecs and connective tissue papillae. Then we can also see these glands in what we call the mucosa. That's the esophageal cardiac glands. Okay, that's esophageal cardiac glands. They are also compound tubular glands. Then we also have what we call the glands, which are now this is the muscularis mucosae. So muscularis mucosae is separating the mucosa, the lamina propria, from the underlying dense irregular connective tissue of the submucosa. Okay, so beyond that, this muscularis mucosae, we have what we call submucosal glands. And these submucosal glands, yes, also compound tubular alveolar or compound tubular glands, okay, which you find them over here. Okay, so essentially they are going to secrete some kind of mucus to neutral mucus to actually nullify or neutralize the acidic chyme, which may actually get closer to the lower portion of the esophagus. Okay, so that is what I want to show you. Now again, for the duodenum, I told you that we are going to have duodenal glands proper. Now you can see these finger-like or leaf-like projections that you find over here, and these ones are what you call villi. Once you can see villi, no one should tell you that the epithelial lining is simple columnar epithelium with goblet cells, because villi is exclusive to what we call the small intestine. But you can see that yes, we are going to have these glands over here, and these represent the crypts of Lieberkuhn. And below that, we are having what we call the muscularis mucosae of smooth muscle over here. And below that, we're going to have submucosa. But within the submucosa, we can see these glands. Okay, these glands. And these glands are known as the Brunner's glands. Okay, that's the submucosal glands or the duodenal glands proper, which you can find in the submucosa. Okay, so that's the duodenum. Now, there are some modifications that we find in what we call the mucosa. There are some modifications. So it presents with something we call folds, okay, folds in the mucosa. But these folds in the mucosa are sometimes contributed, or its core is formed by what we call the submucosa. So let's see some of the folds that we find over there. For instance, the esophagus. Now, the esophagus is always collapsed. So usually if you take a section of a collapsed esophagus, you may see some folds in there. And that's not what we are really looking at. So we say that esophagus actually doesn't have a fold, although yes, usually collapsed, and you may see some folds in there in the lumen. Then for the stomach, you will see rugae. Singular form is ruga, and ruga is the only temporary fold we are going to talk about. Now, the remaining folds we are going to talk about are permanent folds. So, ruga or rugi are transient folds that we find in the, mucosa of the stomach, and they actually form from, submucosa from mucosa and submucosa forming its core. And of course, it will stretch to allow for maximum filling of the stomach. So as the stomach gets filled with food, then the rugi gets ironed out. Okay, they flatten out to accommodate the increasing, food product in the stomach. Then for the small intestine, you may see another fold which you call plaque circularis, and plaque circularis is again mucosa and submucosa forms it. Then for its function, what will happen is that it's for increasing absorptive surface area. Okay, increasing absorption by a factor of 3. Now, plaque circularis is exclusive to the small intestine. Again, we have villi exclusive to the small intestine, having what we call epithelium and lamina propria, increasing absorptive surface area by a factor of 10. Now, when I talk about small intestine, I'm talking about duodenum, jejunum, and ileum. Now, something which is peculiar to both small and large intestines are what we call the microvilli or the striated border. Okay, which we say that you're going to, a surface modification of the surface epithelium. So it's formed from only the epithelium, that simple columnar epithelium, and it's going to increase the absorptive surface area by a factor of 20. So if I have a segment of the small intestine, for instance, and I'm having both plaque circularis, villi, and microvilli or striated border, then what will happen is that I'm able to greatly increase absorptive surface area by a factor of 600. Now, one thing that I want you to know regarding this plaque circularis is that the proximal part of the duodenum, although we are saying that it's peculiar to the small intestine, the proximal part of the duodenum is devoid of it. Okay, just the first part of the duodenum is devoid of it. Now, the remaining part, the distal part, okay, second, third, fourth part of the duodenum will have it. So as you move this way, okay, from the first part to the third part or to the fourth part, then the plaque circularis continues to develop. By the time it gets to the jejunum, it is fully developed. So we say that plaque circularis is quite prominent in what we call the jejunum. Okay, but beyond the jejunum, then it begins to also becomes, reduced in terms of development. Okay, it becomes less prominent, and then eventually it fades out, getting to the distal end of the ileum. Okay, so it means the proximal part of the duodenum as well as the distal part of the ileum are devoid of plaque circularis. Okay, so that's what we are going to see. Now, the rectum presents what we call transverse rectal fold or plaque transversalis recti. Okay, having its mucosa and submucosa forming this component, and it will be very important. Yes, we appreciate that it will be important in supporting the weight of the fecal matter in the rectum as it is being stored. Now, so these are the rugi. You can see these folds. Now, if we talk about rugi, in fact, even the vagina will have rugi, even the palatine ridges, okay, the hard palate, yes, they are rugi. So it means that we have to actually qualify this rugi. So we say gastric rugi because it's related to the stomach. Okay, so these are the folds that we are calling it rugi. They are transient folds. So it's formed by, of course, mucosa, and then the submucosa forms its core. Okay, so that's what you have to know. Now we also have what we call the crypts. Okay, now we said that for crypts, okay, that's the mucosal glands. I've showed you already, but let me just say one or two things about it. Now, the crypts, you can see the Paneth cell, okay, will be at the lower portion of the gland. Okay, so I've given you some notes regarding these Crypts of Lieberkuhn, which I've already explained to you before. Yeah, so lower portion of 20% of the epithelium, so the lower portion, that's where you're going to find the crypts. Okay, and then you can see the Paneth cells. Paneth cells will also be located at the base, okay, of the crypts, as well as, of course, its undifferentiated stem cells will also be there. But Paneth cells will be important in secreting some kind of ions, IgE antibodies, okay, into the lumen. Okay, now crypt cells are very important. They are turned over yes, Crypt cells will migrate, okay, 3 to 8 days onto the surface epithelium, okay, for rapid, repair. Okay, that's one important thing. Although what we say is that they are sensitive to what we call, radiation therapy and chemotherapy. This is what we call the Crypts of Lieberkuhn. The Crypts of Lieberkuhn is exclusive to the small and large intestines. So again, we said that we have the villi. The villi is also here. So that's the villi. The villi is having these finger-like projections over here, and it's formed from, of course, the epithelium and its lamina propria. So that's the villi. So within the villi, you can see the villi is in the small intestine. So you can see simple columna with a lot of goblet cells. So the epithelial lining as well as its lamina propria will form its core. Okay. Then we also have what we call microvilli. Microvilli will also be here. So microvilli actually looks like, these kind of elongated columns over here. Okay, under electron microscopy, this is an electron microscopic specimen that we find over here. Okay, now there's the goblet cells over here. You're supposed to have what we call the striated border. Now, if it's under light microscopy, you call it striated border. But if it's under electron microscopy, you are going to call it brush border. Under electron microscopy, we're going to call it microvilli. And light microscopy, if it's in the GIT, in the small intestine or large intestine, we call it striated border. But if it is what we call the proximal convoluted tubule of the kidney, then we are talking about brush border. Although we tend to use all these ones interchangeably. Okay, that's what we see. Then we also have what we call the Crypts of Lieberkuhn. Now, we're going to have what we call the plaque circularis. For plaque circularis, yes, it's going to just be like that of the stomach, that of the gastric rugi. Only that to bear what we call, villi will be seen there. The epithelial line will also be simple columnar with goblet cells. Okay, will be seen there. Now, one thing that I have to explain to you over here is that, yes, we say that, yes, both the small and large intestine will have what we call microvilli. I want to show you this. Now you see that this is the microvilli or the striated border. Specifically, this is a light microscopic specimen, and therefore it's supposed to be striated border. Okay, so because we are having only striated border in the case of the large intestine, then we say the lumen of the large intestine is almost smooth, or a smooth luminal margin. But for the small intestine, because of these finger-like projections, which may have different lengths and all that, yes, gives it, a serrated lumina, and therefore what happens is that we say that the villi ensures that the lumen of the small intestine is not smooth. Okay, that's what I want you to know. Now, the transverse rectal fold, yes, will be the fold, yes, formed from, of course, mucosa and the submucosa forming its, core. And that transverse rectal fold is exclusive to the rectum. Now the next coat, now we've seen mucosa made up of epithelium, lamina propria, and muscularis mucosae. Then we've seen the submucosa. Now the next one we want to see, where is the next coat of the post-pharyngeal gut, is the submucosa. Is muscularis propria or muscularis externa. Now muscularis propria will have two components, just like the muscularis mucosae, inner circular layer as well as outer longitudinal muscle layer. Okay, so that's what we are going to see. We will see all those functions. Now, what we find is that in the stomach, we are going to have 3 layers instead of 2. Now, the 3 layers are, yes, inner oblique, middle circular, outer longitudinal. The stomach is endowed in an oblique layer as well. Now, bear in mind that, yes, this muscularis propria or muscularis externa we are talking about in the post-pharyngeal gut will be made of what we call smooth muscle, except that areas which communicate with the external environment. The esophagus, as well as, of course, the anal canal. So, for instance, we get to know, the divisions for the, anal canal, for instance. So, this is the stomach musculature. Okay, for the stomach, we are going to have inner oblique, middle circular, and outer longitudinal muscle layers. That's what we find in the case of the stomach. Okay. Now, for you to know, for instance, I talked about, the rectum, the anal canal. The anal canal will be made of skeletal muscle, because communicating with the external environment. For the esophagus, there will be some elements of skeletal muscle and smooth muscle. Now, for that matter, the esophagus will be divided into three: upper third, middle third, and lower third. The upper third, because more closer to the external environment, will be made of skeletal muscle. So this is how the skeletal muscle will look like. If the section is made against the orientation of the muscle fibers, there will be those kind of pieces that you find over here, and these cells are multinucleated. These are the myocytes. They are multinucleated, and the nuclei are peripherally placed, and there's a longitudinal section, okay, going through the muscle. So you can see that there's what we call cross striations as well, okay, light and dark bands in there. So that will be for the upper 1/3 of the esophagus. Then for the lower 1/3 of the esophagus is exclusively smooth muscle. So you can see the spindle-shaped nuclei, which are actually mononucleates. Okay, you can see this for the transverse section running, against the orientation of the muscle fibers. There's a longitudinal section. Actually, muscle fibers, how they are running was how the section was made. Okay, so that is what we see. So the nuclei are centrally placed and they are mononucleate for smooth muscle. Then for the middle 1/3 of the esophagus will be a mixture of the skeletal muscle and smooth muscle fibers. Now, one key thing too that we find, okay, we saw there was a nerve plexus in the submucosa known as Meissner's nerve plexus or submucosal nerve plexus. Now, which was giving some kind of innervation to what we call the muscularis mucosae. Now, we have inner circular and outer longitudinal muscle layers. In between them, these muscle components will be another nerve plexus, and that is what we call myenteric nerve plexus. Myenteric, myen has to do with muscle. It's also given this eponym which is called Urbach's nerve plexus. Urbach's nerve plexus. So it tells you that they will control the contractile activity of what we call the external muscle components, inner circular and outer longitudinal muscle component. Now, I will tell you that yes, you see that the wave-like contractions that we tend to talk about regarding peristalsis of what we call the post-pharyngeal gut is actually coming from the inner circular muscle layer. We will see that. So here we are, this peristalsis. Okay, so peristalsis is actually mainly contributed by the inner circular muscle layer. Okay, so that what will happen is that the adjacent segment of the alimentary canal, okay, post-pharyngeal gut in this case, yes, contracts and relax, okay, to move the product along, the post-pharyngeal gut. So what we are saying that peristalsis ensures propulsion of the food product along the alimentary canal. Okay, apart from helping in peristalsis, what we are saying is that the inner circular also helps in formation of sphincters. Talk about pyloric sphincter, talk about the ileocecal valve. All these sphincters will actually be produced from thickness of what we call the inner circular muscle layer. Okay, so that's what we see. Then what will happen is that for effective mixing of the product in the lumen of the post-pharyngeal gut, then the outer longitudinal is very important in that manner. And that's what we call segmental contraction. Okay, segmental contraction. Such a non-adjacent segment of the alimentary tract or the post-pharyngeal gut, yes, contract alternatively. Okay, yes, actually, to move the food, okay, forward and backwards to help in mixing of the food. But one thing that you have to know is that yes, although we are saying is that this one, the outer longitudinal, is going to play that role. Yes, peristalsis, inner circular is going to play that role. What happens is that they work in synergy actually. But when you see the colon or when you see the cecum, now histologically there's no distinction between cecum and colon. So whenever I say colon, it includes cecum. You have thickness of what we call this outer longitudinal muscle layer, forming what we call TNA coli. Now these TNA coli are three thickened, bands of the outer longitudinal muscle layer. We will see that. So that's what we see. Yes, these are TNA coli. Yes, 1, 2, and 3. Thickening of the outer longitudinal muscle layer. And they are very important in producing what we call the costrations or the circulations that we find with the large intestine. Okay, so that's what we see. So there's a part of the TNA coli. Now, the outermost tunic of the organs of the post-pharyngeal gut, okay, is known as serosa or adventitia. Now, this serosa or adventitia is made of loose connective tissue or areolar connective tissue. Okay, loose connective tissue, areolar connective tissue, adventitia. But if you are having a thin layer, okay, or simple squamous epithelium or mesothelium lining the outermost component of the adventitia, then we call it serosa. So the intraperitoneal organs of the abdomen will have serosa. The extraperitoneal or retroperitoneal organs will have adventitia. Okay, so that's what we are going to see. So, for instance, if you take the esophagus, the post or the pre-abdominal portion will actually be made of adventitia because they are extraperitoneal. But that portion, the distal just 1 to 2 centimeters that we find in, of course, the abdomen will be intraperitoneal and therefore have what you call serosa. So that's what I have. Yes, esophagus, yes, mainly will have adventitia except the distal 1 to 2 centimeters portion. The stomach will have serosa, intraperitoneal, duodenum, mainly adventitia except just a portion, small portion of its first part. The proximal part will have what you call serosa. Now the jejunum and ileum, jejunum ileum, yes, will have serosa, intraperitoneal. Appendix, transverse colon and sigmoidal colon will have serosa. Cecum, ascending colon, descending colon, and of course rectum will have adventitia as well as the anal canal. So if you have adventitia, then you are, non-mobile. You are fixed to structures, surrounding structures. But those ones which are mobile, having serosa, okay, will actually be mobile, actually. Now, to end this whole presentation, now what I want you to know is that there are some changes in epithelial lining. For instance, between the esophagus and the cardiac portion of the stomach, okay, we are going to have the gastroesophageal junction or esophagogastric junction or esophagocardiac junction. Okay, so you know that the esophagus communicating with external environment will have to be protective. Therefore, it's having stratified squamous, non-keratinized epithelium. Now, at the point when it gets to the stomach, changes to become what we call simple columnar epithelium without goblet cells. So this area is what we call the squamo-columnar junction, where squamous epithelium changes to columnar epithelial tissue. That's what we're going to see. Okay, so there's one important thing that we can tell to distinguish between these ones. Okay, so these glands, okay, that we find here represent the esophageal cardiac glands that we're talking about. Okay, so there's a real-time section showing you there's the esophagus and this is the stomach, showing you the gastric pits and all that, as well as of course the gastric glands will be there. Okay, that's stratified squamous, the simple columnar without goblet cells, but you can see a lot of mucinous substance over here, pale staining area. Then we also have what we call the the gastro now this is the parietal part of the stomach and this is the duodenum. The duodenum in its submucosa are going to have what we call duodenal glands proper, known as Brunner's glands over here. And then the duodenum also have finger-like projections, with its epithelial line being simple columnar epithelium with goblet cells and brush border, or striated border actually. Then of course, the pylorus of the stomach, okay, will have what we call simple columnar epithelium without goblet cells, okay, which you see over here. That's what you can see. So there's a change in epithelial lining this way. Okay, now remember that there's no sharp boundary. For instance, if you look at the duodenum to jejunum, okay, but you know that the jejunum will be devoid of submucosal glands, but the duodenum will have submucosal glands. That's the distinction. Again, you can't really, there's no sharp boundary between, of course, the ileum and the jejunum. Okay, but what you can say is that, of course, the ileum will have Peyer's patches, okay, which you saw in its mucosa, sometimes in the submucosa, but you don't see it in the case of jejunum. Okay, for instance, if you look at the cecum and then the ileum, what will happen is that yes, the cecum will have a smooth luminal margin. Okay, it also lacks villi because of lacking villi. Yes, of course, if you take the cecum will have, both of them will have goblet cells, but you know that Peyer's patches will be seen in the case of the ileum. So that's also another distinguishing feature that we see. Okay, so there are several ways that you can go about these ones. Now, let me show you the real-time section for the duodenal junction. Gastroduodenal junction. Now, this represents the simple columnar epithelium with a lot of goblet cells in the epithelial lining. This is the intestinal portion. Therefore, this is the duodenum. This is the muscularis mucosae. And then below that, the submucosa. You can see glands in the submucosa. These are the duodenal glands proper. That is the Brannus glands. And then these are, these represent, the stomach epithelium, simple columnar without goblet cells, showing you the gastric pits as well as, the gastric glands, in its mucosa, So that's what, we're going to see. Yes, showing you the anorectal junction. The rectum will have simple columnar epithelium with a lot of goblet cells, and then the anal canal, will have stratified squamous epithelium stratified squamous non-keratinized epithelium. You can see it over here. So it means that there's another squamocolumnar junction, over here. But I told you that the muscularis mucosae, in the rectum beyond the anorectal junction, which is over here, is actually deficient. We don't really see it. So we say muscularis mucosae is absent, beyond the anorectal junction. That's what we see. But you can see that the muscle, you can see the internal anal sphincter, which is actually involuntarily controlled. And then you can also see and therefore made of smooth muscle. And then you can see the external anal sphincter, made of actually skeletal muscle, controlling the ejection of fecal matter. Of course, levator ani in the pelvis. So please visit the video I've done in the pelvis and perineum. Actually, in the perineum, you can see levator ani muscle over there. So we see all these ones. So there's actually a real-time section showing you, from the rectum. Having, there's a stratified squamous non-keratinized portion, which is actually the anal canal. That's the anal canal, and then we get to the anorectal junction, the squamocolumnar junction, and then you also have what we call the simple columnar epithelium with a lot of goblet cells actually moving this way to this portion. So you can see the Crypts of Lieberkuhn is also being there. So this is what I have for you for today. Just know that for the post-pharyngeal gut, instead of having the normal 3 coats or 3 tunics, it's going to have 4 tunics. And these are the mucosa, submucosa, muscularis externa, and adventitia or serosa. I hope you find it helpful. Please do subscribe to the channel so that anytime I upload quite interesting videos as these, you will be able to be the first to watch. Have a good night, and may God richly bless you.